Hello, and welcome to another episode of Thoroughly Thriving, where we're putting human back in human capital solutions. On today's episode, we're going to hear from an industry leader where they're sharing their thoughts, feedback, and perspective on what's happening in today's world of work. Well, let's stay tuned and perk our ears up for another exciting episode. Well, welcome to another episode of Thoroughly Thriving. And Sean, I'm really excited for this episode. We have a pioneer within life sciences joining us on the podcast today. And our guest today has spent nearly 3 decades doing something rare in our industry. And we have Bassil Dahiyat from Xencor joining us. And Bassil has spent the last 3 decades building one company, which is rare in our industry. And he's done this with conviction, from an idea into a clinical stage biopharma with multiple marketed drugs to its name. Well, Dr. Bassil Dahiyat is the co-founder and president of Xencor, a company that he started back in 1997 with a bold premise that you could, in fact, engineer proteins, design antibodies the way an engineer designs a system. This is to create better medicines for patients with cancer and autoimmune diseases. Well, that premise became the XmAbs platform, the technology behind a pipeline of engineered antibodies and 3 drugs that have reached the market through Xencor's partnerships. Well, Bassil is a scientist at his core. He holds a PhD in chemistry from Caltech and biomedical engineering degrees from Johns Hopkins. He's inventor on more than 60 patents and co-invented the XmAbs technology himself. He's been recognized as a top 100 young innovator by MIT Technology Review, and also recognized as a technology pioneer by the World Economic Forum and the EY Entrepreneur of the Year in the Los Angeles region. But what makes his story so compelling, listeners, isn't just the science, it's the leadership journey behind it. Founding a company, stepping back, stepping forward again, and building a culture and a team capable of sustaining a long, hard scientific bet. Bassil, welcome to Thoroughly Thriving.
Thanks a lot, Darren. It's really daunting when I hear that phrase. 3 decades of working at the same company, and I always ask myself, "Gosh, that's a long time. Like, what have I gotten done?" You know, I've got to look back at that and say, "Geez, there's still so much more to do." But it's been really rewarding and exciting over the years, even wrestling with the various downs that come with the ups.
Well, it's said like a true innovator and a leader and a pioneer in regards to saying there's so much more to do. But I'm looking forward, I know Trish and I are looking forward to learning more about your journey and your perspective and your leadership approach and the culture that you've helped create at Xencor. So Bassil, let's jump into it. Xencor was built on the idea that proteins could be engineered, designed deliberately rather than discovered by trial and error. If we go back to 1997, that was far from accepted back then. For our listeners, Bassil, that aren't protein chemists, what does engineering an antibody actually mean? And why did you believe back in 1997 that it would work when many, and I mean many, didn't think it was possible?
Well, in fairness to them, it took quite a number of years to get all the pieces in place and the tools to work together so that we could make useful, impactful molecules that could be medicines. But the vision we had was based on working in a very sort of a very cutting edge area at the time. Now it's old hat. It's applying computational methodologies to biochemistry, in particular, looking at how the structure of molecules, how the structure of proteins in particular drives their function and how you could potentially manipulate that structure to manipulate the function, right? So these are really, really nanoscale machines built out of amino acids that take on the shapes that the natural code of amino acids gives them, and they do jobs. And our idea was we can manipulate those machines. And, computational methods were very, very, very much in their infancy back then. We were excited by having 16 megabytes of memory in our processor cores. We were thrilled when it went to 64 megabytes. And so the idea was that we could automate and use rules, particularly biophysical rules, to help an algorithm make the decisions about what should take place. So proteins are just long strings of amino acids. The amino acids, there's 20 different commonly naturally occurring ones, and they all have slightly different physical and chemical properties. And so a string of these put together in order, different amino acid orders, lysine, alanine, phenylalanine, tryptophan, is going to create sort of this chemical code. And this creates this natural folding up of structure. I always find that incredibly fascinating, that just stringing together these beads on a string, these amino acids in order, there was information embedded in there, and just you put it in solution and it folds up and it makes automatically a machine. So I loved that. And we were the first to demonstrate that you could automatically design an entire protein within a computer without human intervention or bias. Just say, here's the algorithmic rules based on biophysics, go, right? Now, that was a toy protein. It was only 27 amino acids. It didn't actually do anything. But that was the concept that got us excited and said, there's potential here. And that's sort of a, pretty ill-informed at the time imagination about what you could accomplish with it was what the drive was to start the company, with my thesis advisor and I together. And, we got people excited about that, and we're able to generate initial investment capital, a small amount, $1.5 million, from an unorthodox group of investors. They came out of the financial services world, but they were very interested in cutting-edge university technologies. And they financed a company for DNA diagnostics out of Caltech a few years before.
Wow.
And so we were just these young Caltech guys with this idea of making new protein sequences. And the buzz of the industry at the time in the late 1990s was Amgen and Genentech, and they're discovering genes and they're patenting them, and they can capture the value of those while they make important medicines like erythropoietin and human growth hormone. And so the idea was you can make new sequences. Those are new patents, right? The patent office recognizes biological sequences. Wow, that could be really valuable. And of course, that has been really valuable. But the way it's played out has been much more, challenging to predict and meandering, like a river delta going rather than a straight shot, right? And so all we do really is use computers to decide what order of amino acid beads to put together. Or if you've got a big protein that already exists out there, like an antibody, where to make changes to make it do just the job you want. And the funny thing is, when we started the company, we wouldn't have dreamed of working on antibodies. Antibodies were considered a failed drug class in the late 1990s.
Wow.
Right? All the initial ones just didn't work. So that's kind of the scientific setting is you've got this great tool. You can create new patents. You can sequences. You can patent them. What might you do? And that was in a nutshell, the challenge of Xencor. What are you going to do with this?
It's just such an interesting story around the idea of conviction, right? The idea of, we're convicted that this really has legs and let's put something behind it. Your journey, Bassil, has been a pretty interesting one from founder. You moved from CEO into CSO, the Chief Scientific Officer role, in 2003, but then returned as the CEO in 2005. And so from the outside, some of us observers would have some questions like, what did that transition teach you about some of your own strengths and opportunities for growth? But also, what would you say to a technical founder right now, maybe who's listening to this, who's wrestling with, am I the right person to lead the organization?
So for me, we were, at it for about 4 years or so. We'd accomplished some really critical goals. We'd figured out a lot of the limitations of the initial tools we developed at Caltech for how to work on more meaningful proteins. Hadn't quite figured out what exactly to do. Do we want to make them longer half-life? And if so, how? Do we want to make better binders against new things? Do we want to manipulate? What do we want to manipulate biologically that's going to make an important medicine? But we'd also kind of leveraged a very opportunistic financing market in 2000, the dot-com bubble, which, AI is pretty hot today, but boy, the internet was really hot in 2000. And we leveraged that into raising a lot of capital. We raised a $50 million round. I mean, think about it. In 3 years before that, we raised a $1.5 million round. That's when I learned that the financial world changes very rapidly. And of course, within 8 or 9 months, that whole dot-com bubble collapsed, became the dot bomb. And we were really thrilled to have the money. Then we could really invest in building the company, really where the number one thing we were building was finding people with expertise that we lacked. People who understood a little bit about drug development, about drug discovery, what the needs were for new medicines. And it helped us broaden ourselves. And what it really made me and the board of directors, our investors, because your board when you're a small company like that is your investors, we're like, gosh, we're missing a lot of perspective. We're hiring these people. It's really changing how we're thinking about our science. We're still trying to figure out what really to emphasize with our work and how to position it in a way that we could find partnerships and generate. Essentially, the theme at the time was very much more about create a business, not just make your own drugs, right? The industry's changed a lot since then. And we weren't getting much traction with pharma partners. We'd done some industrial enzyme deals, some ag chemical trait deals, and those are very low margin industries, right? Soybeans, corn, right? Butadiene. This is not like medicine. So it was like, well, we should bring in somebody who understands what a deal looks like in our industry, what captures excitement and interest, and what are the right directions to move in. And I think the thing I discovered about myself at the time, which is a strength that I try to lean into, is I'm pretty objective and analytical. Mm-hmm. So the first thing that comes to mind when something new comes at me, like our main investor saying, I think we got to bring in a CEO to tell us what the heck we're going to do, really, with this great stuff you're creating. And my first reaction was, you know what, that could really solve some problems. It wasn't an emotional one. And I think, that's something that I sort of inherently believed about myself, but it really came to the fore then. I was like, okay, yeah, this is good. What can we make of this? How can we make this successful? How can we find somebody who can really change things? So being analytical, being objective, and trying to look at a problem from different directions is a strength of mine. And so that's something I definitely try to lean into and always keep at the forefront, because we're in a technical business, and being objective and analytical about things is a very, very important aspect of that. And so, I said, yeah, let's try to find somebody. The challenge was, one, it was a very small industry at the time relative to now. Two, we hadn't really done much other than some cool science. So we really weren't able to find the world-class leaders yet. Because one, there were very, very, very few in our industry who'd really developed any chops. And so you were always taking a risk, right? It was a much smaller place back then. I mean, San Diego was a handful of buildings up on the mesa by Torrey Pines, not this, sprawling mass that it was now. And anyway, we one of our board members knew somebody who knew somebody. We had some interviews, sounded like he was really confident about how you engage with pharma partners, how you do deals. And so we brought him on. And, I think I had misgivings from the beginning. Is this the right person? They don't seem to be a fit with the things of value that we can create, that we could actually deliver, right? They're coming up with different ideas, but I'm like, I don't know that we could do that, right? I don't know that anybody could do that, or maybe somebody could do that, but it has nothing to do with what we're doing. So if you want to just change the whole company around, maybe that's fine, but I'm not connecting the dots here. That's not what you're saying. So I was initially went, but we gave it a try, and it lasted about 18 months, and it became very clear that both from a sort of fit and style and culture standpoint of know what you're doing and then aggressively pursue it and then evangelize to find the partners and people that can invest with you, it was more a matter of, okay, what do I think people want to hear? right? Let's match to that. And that never works in a technical business. I mean, maybe it does if you're like, I don't know, writing apps or something and you can make a new product in 3 weeks. Maybe, I don't know. It doesn't work in our business.
Wow.
And, that dissonance from the vision that our investors had pretty quickly led to, this guy isn't working out. And it was sort of like we're stuck with Bassil, so let's go with it and see what we can do. And we started building from there. And, actually we brought in we added, I should say, to our initial group of high net worth individuals coming out of the trading world, I should say, in 2000, about 4 or 5 years later, 3-4 years later, 2006, institutional VCs from the East Coast. And their first thing was, wow, we always, find a professional CEO to put on top of the scientific founder. And they searched for a year and couldn't really find anybody that even got them excited. Because again, we had started to make some traction, started to do some deals, had decided on antibodies is what we wanted to work on then. And, did a number of partnerships, small ones, but exciting ones, generating revenue with companies like Genentech and Chugai and Pfizer and MedImmune, top names. And yet still it was like, wow, who's the right person? We had a nascent pipeline. And so it just didn't go anywhere.
Well, we're going to pivot a little bit we're going to build off that question around leadership.
Yeah.
But we're going to weave in and we're going to talk about Xencor's pipeline a bit. With pipeline across oncology and autoimmune, major readouts and a pivotal study ahead, how do you take and make the high-stake calls about which programs get resources and which timelines hold? And how does that decision-making change since those early days that you just talked about?
I would say that you have to be very, very thorough and analytical. There's that word again in looking at what you've got, right? What data do you have? What data might you get if your thesis plays out? How do we see the competitive landscape working? And the competitive landscape isn't limited to people doing the kind of thing you're doing, like, oh, you're making an antibody against target X, and somebody else is making an antibody against target X, and somebody else is making an antibody against target X. And let's look at those two competitors and what their product features are and how far along they are and how much financing they have, important parameters, right? How robust of a competitor they're going to be. That's just like the start of it, right? You've got to open your mind and be creative. There's competitors that are doing things completely different from what you're doing with completely different tools, but they can solve or make less important the problem that you're working on solving. And then nobody's going to need your solution, right? And so that thorough analytical piece has always been constant. I think the thing that's really changed since the early days and it's sort of changed gradually and then with a steepening slope over time, is bringing in people who have expertise and experience to be thought partners along the way and really listening to them. I mean, that started in 2000 when we were able to go from just some, postdoc type people designing proteins and expressing them and purifying them and characterizing them to people who'd actually worked on drug discovery programs, people who'd put molecules in the clinic. So bringing in expertise in that way and listening to them, and that's grown over time. But in particular, outside expertise, right? The physicians, the big pharma people who've launched drugs in these areas, regulators and former regulators, really digging in with them and having them point out this, that, and the other issue, and trying to keep all that information together. Because if the people who work all day wrestling with the problem that you're trying to solve, whether it's, a Hodgkin lymphoma or rheumatoid arthritis, if they can't really get excited about the solution you're providing, you don't have a solution, right? So you've got to have those experts and that expertise. Experience and expertise matters, right? Demonstrated track record matters. They know the pitfalls. You've got to listen to them. So that's something that I was much more internally driven 20 years ago doing the analysis, thinking about things, gathering information, rather than really bringing in others and seeking them out, right? And trying to figure out who's the right group of people for this aspect of the question, the regulatory aspect of the question, or the clinical development aspect of the question, right? Right? Or the safety aspects of this program. That's been a big change, right?
Wow.
You got to have the right people around you, and you've got to excite them.
Yeah.
It's so interesting. And I think to your point about the experience value, we're seeing as we work with a lot of small and emerging companies, the gaps when you don't have it or you don't have leaders willing to pursue it, that causes challenges in being able to ultimately achieve the goal that is ahead. So I think it's a great way to think about it. And, you talked about, continuing to kind of grow and pressure test that in each individual situation as being different, right? To your point about, that this experience is more important for this problem we're trying to solve, or this specific program, or this specific stage, or whatever it may be. I want to kind of pivot a little bit into my next question that kind of builds on this around the importance of culture. Engineering-driven drug discovery has oftentimes a pretty slow feedback loop that can be measured in years. I had somebody when I first got into this space with working with small and emerging companies that said, Sean, you'll never make it if you don't realize that your definition of fast and my definition of fast are two completely different things. That the feedback loop is not weeks, it's not months, it's often measured in years. So how have you found is the best way to develop a culture around things and to keep your team really motivated and rigorous throughout long periods of uncertainty? And how do you continue to protect that culture as you've seen Xencor grow over the last, many years?
I mean, my founding investor, who, was a close colleague for, about 2 decades before he sort of, exited a lot of his position, moved on, semi-retired, always used to say, why does everything in this business take forever? Nothing happens, right? Years go by and we still don't have a drug. Well, what is this business, right? Yeah. So what you're saying is very, very true. I think our entire enterprise, our culture, the people that thrive in it, it's based on doing bleeding edge science and trying to apply it to practical problems. In our cases, in molecular design problems using proteins, right? So that's the core of us. And so we just try to stay true to that, right?
Yep.
Even if the majority of our spending and time in the day of the leadership is spent on working on this clinical trial or that one, that core thesis we maintained. And I think that's very, very important. And that's why we've had this 30-year continuity. We are always investing in, even if, we had stretches of time during the financial crisis in 2008 to 2011 where there was only about 15 people working on it, wow. Right? By the way, nearly all of those 15 people are still in this building right now that I'm sitting in, working on proteins that they're designing. We're always saying, but we are going to try to do the next thing, right? You can have that bleeding edge work going on while you're advancing molecules, while you're consolidating, gaining knowledge. But that's been really the key. And, I've had to be very, very, very sort of front and center on that, demanding that we keep to that. I've had investor groups over the years say, why are we still spending money on this? Why are we doing this? And I've had to be very, very firm about that, right? Because that's who we are, and that's what gives us a differentiating edge, right? If we lose that, maybe you can thread the needle and succeed with what you've already created. Maybe you can't, right? A lot of companies do, right? In fact, most companies in our business only have that thin thread of one or two things. And if it gets clipped, if that technology thread that they're hanging on gets clipped, there's no more them, right? And that might be fine. That's a way to build an industry and a career that's based on this, I'm going to take a narrow focus. It fails, it succeeds. We always felt like we had a purpose that was kind of bigger and beyond us, of advancing the real cutting edge science and using it, again, to engineer molecules, which is now old hat. People talk about it all the time, but there's still amazing new work to be done And we're still pushing frontiers today. And so that's where you've got to have your own convictions and where you've got to be the leader and say, no, I'm going to put my reputation on. I'm going to stake my career on it. And we're going to push this even when people push back. And, I don't want to make it sound like in the prior answers to the questions, like whether it was when we had, another CEO come in or when we were making high stakes decisions, that I'm not front and center and being the person ultimately who's responsible and taking difficult positions sometimes, because no matter how many great experts you have, there's no perfect answers because they can't read the future either. They're just giving you better analysis. So you've got to be able and ready to just say, okay, enough. This is as best we're going to analyze things. This is what we're going to do based on all of these facts and then all of these assumptions I'm making, because that's the best I can do. Right? So I do try very hard to articulate the why of a decision, but that's a decision. So, I don't want to ever make it seem like I was disengaged from it or just a participant or a spectator. Like when we were searching for new CEO, I was front and center saying, no, this is why this person is not the right one. Yes, this one maybe, let's see, right? So I just don't want to imply that you can go get by just being sort of passive and letting the world sort of waft you along on its wings. No.
Bassil, a recurring theme on our show has been failing forward. A little alliteration there. But over 30 years, what's a program or a decision that didn't go as planned? And how does it reshape the way that you lead or the way that Xencor operates?
I would say, thinking about that, something that's really shaped a lot of how I think and operate is, this is going to go way back almost 20 years. Our very first molecule we put into the clinic ourselves. And we did try to do our own drug development in the late 2000s. So we had our monoclonal antibody we created called XmAb2513. It targeted CD30, and it was going to be used to treat Hodgkin lymphoma. And there have been other people trying anti-CD30-based antibody structures that hadn't worked. We had created our new technology for FC engineering, the bottom half of the antibody that, you think of antibodies as the top half of the Y sticks to some antigen, sticks to a bacteria, sticks to a cancer cell and takes care of it. Well, how does it take care of it? The back half of the antibody, that sort of structural stem of that Y shape called the FC, engages with the rest of the immune system. And so we'd engineered FC domains to engage much more avidly with the killer part of the immune system, natural killer cells and macrophages, so that when you did stick to a target, you brought a lot more killing power. And our first program into the clinic was this molecule against the target CD30 for Hodgkin lymphoma. And we got it going, and we'd gotten maybe a dozen patients in dose escalation and started seeing some responses and patients doing very well, and it was very well tolerated. And we were very excited by this. Like, this is great. Maybe we're going to be a drug development company too. And then a couple things happened in that milieu. One, the financial crisis hit, and where our next dollar was coming. This was 2009. So late 2008, the financial world imploded, and the money in biotech just literally evaporated. And it was a case of, okay, gosh, that's bad. We had to scrape up the money to keep developing this molecule. And then we had a competitor doing something kind of different from us, making antibody drug conjugates that in 2009 had just consistently failed. They'd never worked over and over and over again. So I kind of dismissed them as competitors. Well, like, that was not a good assessment. You don't make the right calls all the time. And, that was based on analysis that I stick by today. It's just sometimes people discover something new that you don't anticipate. Yeah. And so this was shading into 2010. That was a program called SGM35, which is now the drug Etcetras. Mm-hmm. And it was a molecule that was about a year and a half, 2 years ahead of us. And they started showing good results. And there's not that many patients with Hodgkin lymphoma. It became harder to recruit because all the patients were going to that molecule, which had finished phase 2, had a compassionate use, was probably going to get accelerated approval. And we made the decision to not go forward with our program in the clinic anymore. That was a mistake. We thought, the market's too small. That was a mistake. It was based on we're scared of not having any money. This competitor's too far ahead of us. We had a differentiated molecule. It was very well tolerated but active. It could potentially be combined with chemotherapy. The money to develop it in these initial next phases was not enormous. Very difficult to get at the time. But, I didn't stand up on the table at the board meeting and say, we have to do this because we've got a winner here. We just have to keep going, right? We let it go. So I deeply regret that. And I still think to this day, we would have had a great drug if we'd pursued it. And it's made me not let the idea that, gosh, I'm not sure, I don't know how we're going to do this, get in the way of good decisions, because I could look at the data, look at our product features, look at our competitors' product features, look at, how it might play out over the next couple of years, how a partner might see it, could we finance it that way in a couple of years? All we had to do was get through this. I sort of let this sort of general flow of my board and investors and the downtrodden zeitgeist at the time in the financial world make the decision, which was bad. It wasn't based on the science and the analysis. So, because we've canceled a number of programs after that we put in the clinic over the years, because we really had a hiatus of doing our own internal drug development until about 2016. We out-licensed other stuff and just hunkered down as a tech company. And who knows what the path could have been if we hadn't gone that way, right? Again, I had investors that were dying. They were bleeding out the side of their head. Right? We have no money. Oh my God, what are we going to do? I regret not finding a way.
It's I'm familiar with the other program as well, so it's
everybody is.
Yeah, it's interesting.
They built a great company.
They did. Yeah, but now that's affected so many of the other decisions that you've probably made over the years to say, I'm not going to allow that one to limit. I love how you said I would still defend the reasoning, because I think, who we are, ultimately we've got to be able to still remain analytical. We still need to stick to our core of what it is that we utilize to make decisions, even if the occasional one doesn't lead us to the place we want it to be.
Yeah. I mean, look, the premise of that program was based on a wrong analysis by us, which was antibody-drug conjugates are never going to work because they've never worked before. The past doesn't predict the future, et cetera, right? We've all heard that.
That's so true. All right, well, I want to leave us with our last question here. For a lot of our listeners, we always like to leave something practical on our episodes. I have learned a ton over the last 30 minutes plus, and I'm sure all of our audience has as well. But I want to leave something practical for the scientist, who's dreaming of building something really durable in life sciences. Like, I'm sure in 1997, you hadn't quite foreseen that this is what 2026 would look like. But
no.
What's some advice that you would give, kind of that early stage, scientist or leader, inventor, whatever it is, who is kind of dreaming of building something? And maybe if you want to add on to that, maybe one thing, maybe it's the same thing you wish someone would have told you in 1997 that would have, helped guide you through the financial crisis and everything else that, that you guys have been through holistically, over the last, almost 3 decades.
Yeah, I would say you've got to have a flexibility in your thinking about what the things you're doing might be good for. Believe in your science, push it to the limit, do the good things that you're good at, but how they might be applied, you've got to be much more flexible in your thinking than you imagine, because the world is big. The problems that need solving, whether it's new medicines in lymphoma or new diagnostics to detect cancer early, right? Or new structures that are going to revolutionize how people make, nucleic acid therapies. Those things are difficult to know when you're in your box doing your technology. And they're difficult for anyone human being to know. So be flexible, get yourself out in the world, hear the people that are trying to solve these problems or that are the end users, so to speak, right? Do your market research, talk to the doctors, talk to the scientists at the other companies, big ones and little ones working on similar things, or the physicians doing the drug development. to see, maybe we could fit there. That's the best piece of advice. Don't abandon your technology. Figure out where it really ought to live. Because that's the hardest part about starting a technology company and having something that's going to be durable and lasting. And you can build a durable and lasting company finding interesting drug molecules from all sorts of places and stitching them together and deliver a lot of benefit to patients and value to investors. And that's a great business model. That's just not what I do and what Xencor does. So if you're going to try to do that technology-based thing, and we are always going to need that, because you're not going to find good new molecules unless somebody dreamed them up and built the tools to make them happen. And the world is not going to advance without that. But gosh, you gotta talk to the world. You gotta get out in the world to see how it's going to benefit people.
Well, this has been extremely engaging and informative. We've had many guests over the last couple of years on Thoroughly Thriving, but none that has had a similar journey to yours in terms of being with one company for close to 3 decades. And I know I speak on behalf of our listeners and Sean in saying, Bassil, we really appreciate your vulnerability and transparency.
Thank you. It's been great thinking about these questions and trying to, answer them in a way that teaches me something about what's happened. And they're great questions, and they've really, really done that. It's been a pleasure.
Well, thank you so much for joining us today. And as we remind our audience at the end of every episode, and I'm sure your teams over the years would echo that you share the same core value that, as we continue to all work together, pointed in the same direction, that as we work as one, we will also win as one. And so we're thankful for you joining us. We'll get all of Bassil's contact information in the show notes for you. Thank you so much for joining us, and we'll look forward to our next episode of Thoroughly Thriving. Thank you for joining us today for another great episode of Thoroughly Thriving. If you enjoyed today's episode, please feel free to share it with those in your world. If you'd like to learn more about the Thorough Group, please go to thoroughgroup.com. And remember that we will all remain thoroughly thriving if we continue to work as one and win as one.